I'm sorry about the slightly off-frame PowerPoint here. When we think about how organisms adapt to the environment, of course we can think about both abiotic and biotic factors. When we think about plants in particular, we can imagine that plants might adapt to differences in soil, nutrients, and water content. They might adapt to different diseases and pathogens, herbivores, various other abiotic stresses, flowering time in response to seasonal cues, for example, different pollinators, and many other environmental factors. In order to understand how plants adapt to their environment, we need to address these issues at multiple scales, from the molecular, cellular, organismal, ecological, and evolutionary scales. So why should we study the molecular basis of plant adaptation? In my mind, there are really two main reasons. The first is a basic one in evolutionary biology. Adaptive evolution leads to new species and increased biodiversity, and so we would like to know what are the evolutionary and molecular mechanisms that underlie this biodiversity. But there's also a very central question in basic plant biology. Natural selection has resulted in an astonishing diversity of mechanisms that plants use to cope with extreme and often unpredictable environments. How do developmental, physiological, and molecular mechanisms that underlie these adaptations work? And ultimately, we would like to translate findings on the molecular basis of plant adaptation to such basic research that in order to improve our food, feed, and fuel. I think that this is a perfect time to develop new plant systems for understanding ecological genomics. The reason for this is that by studying novel plant systems that haven't traditionally been studied in great detail, we can investigate adaptation to new or extreme environments. We might discover novel molecular mechanisms for adaptation. And now that we have increasingly inexpensive sequencing, there's a much lower barrier for genomic investment. And I think it's important to, when you think about developing new systems, to focus on species or genera with wide ecological diversity. There's value in developing groups of related species in addition to wild crop relatives. And ultimately, I think an important thing is that novel plant systems be genetically tractable so we can use forward genetic approaches to understand diversity and adaptation. So we've been working on a group of plants in the genus Mimulus, and for those of you who aren't familiar with Mimulus, it's really a fantastic system for studying adaptation and evolution. There are about 150 to 200 species worldwide. They range, their primary center of diversity is in western North America. Here in California is the heart of Mimulus country. But there's also a very large radiation in Australia and New Zealand. They range from tiny little aquatics that are used as aquarium plants to big woody shrubs here on the coast of California. There's an excellent species level phylogeny, and Mimulus species have been famous over the years for their extreme floral diversity. I'll show you some of that in a second. Varied mating systems from outcrossing to self-fertilization, and adaptation, as you'll see, to very local or extreme environments. The great thing from a genetics point of view is that a lot of this ecological diversity is encapsulated with what are termed species complexes. These are groups of closely related, often interfertile species that can be crossed in the lab, so direct genetic analyses are possible. They're also exceedingly easy to use in experimental settings. It takes about 2 months to go from seed to seed, very rapid generation time. The plants are often small, so you can grow thousands in the greenhouse at any point in time or in growth chambers. A single cross yields hundreds of seeds, and so they're really ideally suited as a genetic system. You can also do field work by taking individuals of known genetic ancestry, or you can generate individuals by crossing in the lab that contain novel alleles, for example, and you can plant them out in the wild as seeds or seedlings and follow their survival in the natural habitat throughout their life cycle. But until a couple of years ago, there really were no decent genetic or genomic tools with which to exploit this diversity. This just shows some of the extreme floral diversity found within the genus. And this slide also allows me to emphasize that the plants can be very tiny. These are tiny desert annuals, less than a centimeter tall, to these over here going off the screen that are large woody shrubs that are, if you can believe it, taller than I am. These are very woody species on the coast of California. Most of the work in the genus has really focused on two different species complexes. This is Mimulus-Vitis and cardinalis. Some of you may be familiar with the work of Toby Bradshaw and Doug Shemsky. There's been work in this group. Most of the work by the other labs, including our own, has been focused on the Mimulus-Vitis species complex. I should say that also developing genomic tools and genome sequences in Mimulus will also help facilitate broader comparative genomic studies in plants. As many of you know, this is a phylogeny of plants, and you can see that a lot of the diversity, especially in flowering plants, is encapsulated in these asterids, this portion of the phylogeny. And Mimulus will actually be the first, as you'll see, the first genome to be sequenced in this very large diverse group. Up here you see Arabidopsis and poplar and other species as well. So there are broader, more comparative reasons for studying Mimulus. A few years ago, we were very fortunate, myself and a number of colleagues, to get support from NSF to begin to develop genetic and genomic tools for these two major groups, the Mimulus-Vitis complex as well as the Lewisii complex. We're really sort of developing from scratch two parallel model systems. Most of the effort, though, has been focused on Mimulus-Vitis. So we now have a large number of VSTs, genetic markers, genetic linkage maps, physical maps based on fingerprinted back libraries, and these have been integrated using Ovigo hybridization. We've recently developed transformation methods using agrobacterium that are successful, and a lot of this can be accessed on our website, mimulusevolution.org. But we were especially thrilled, of course, when JGI decided to sequence Mimulus-Vitis genome as part of their community sequencing program. And fairly recently, there's been a 7x shotgun sequence that's been produced. And the assembly, it's no longer preliminary, our first assembly of all of the data has shown us, rather surprisingly, that Mimulus did not assemble as most other plants have been assembled. It's sort of a problem child from that point of view. What we found is that there were a large number of complex repeats that were sort of these vast sea of repeats separating gene-rich regions. And as a result, our genome assembly, even though we had over 70,000 paired back-end sequences, our genome assembly basically resulted in no scaffolds over 5 megabases, a lot of scaffolds between 5 and 1 megabase. A lot of the genome is encapsulated in those. But still, we have a lot of scaffolds that contain genes. We don't know exactly where they reside on the chromosomes as a result. So right now, with JGI's help, we are genetically mapping as many of these scaffolds as possible to chromosomes using Illumina sequencing of recombinant inbred line populations. We now have over 500,000 ESTs, and annotation is currently underway. And I'll tell you in most of the talk how we've begun to leverage this sequence to understand adaptation and speciation. But I'll first just give you a few sort of highlights from our study of the genome so far. And these are just a few slides because we're still in the process of annotating. So first, in terms of repeat composition, there's, as I said, a lot of repeats, and it's structured in ways that are sort of reminiscent of maize, in the sense that there are large seas of repetitive DNA that separate these gene-rich scaffolds. We have developed fish, and so some labs are beginning to look at the chromosomal distribution of some of these repeat elements. And as I said, we're just beginning to annotate the genome, and this is a screenshot of some of the initial annotation of the genome. What we can tell already is that it looks as if Mimulus, which is an asterid, has, from looking at the Mimulus genome and comparing it to other plants outside of asterids, is that it appears as if there's been an asterid-specific ancient polyploidy event. And we can see this because if we look at grape and compare it to Mimulus, we see for every grape segment, we see 2 syntenic regions in the Mimulus-Vitis genome. But this is a fairly ancient polyploid event that's likely shared with a lot of asterids. One other thing that's very interesting, when you look at other genomes in the asterids that have, say, for example, back sequences, there's clear evidence of nice microsynteny, and we see this even on larger scales as well. But what I'd like to focus on today is some of the ecological diversity that we find in Mimulus and how our lab and other labs are beginning to use forward genetic approaches to understand this diversity. So this is a shot just sort of highlighting some of the diversity within the Mimulus-Vitis complex. This is a group of perhaps 20 named species that are all more or less interfertile. And there's a question as to what are species, what are highly divergent populations, and so forth. But Mimulus-Vitis itself is largely outcrossing, although it's self-fertile. And this is a highly variable species in itself. There's a radiation of relatives down in Chile that have undergone this explosive radiation and flower color patterning, as opposed to these rather boring but lovely yellow guttatus species. There are about a dozen self-pollinating species of Mimulus that are derived from a Mimulus-Vitis-like ancestor. This is one of them, Mimulus nasutus. I'll tell you a bit about that in a second. You can find Mimulus-Vitis at very high elevations, very extreme cold temperatures, harsh conditions above tree line, above 12,000 feet, all the way down to sea level here on the Pacific coast, where it can be splashed with seawater, and there's salt-tolerant plants, as I'll show you. You can go out to Yellowstone, to the hot springs, find plants flowering in very hot soils in excess of 50 degrees C in the winter, one of the coldest places in North America, or the US. Here, the state rock of California is serpentine. Those of you who know about serpentine know that it's a very harsh place for plants to live, and yet Mimulus-Vitis has adapted to it beautifully and is happy as a clam, maybe happier. This is a Copperopolis here in California. Many of you may know this place. There's some abandoned copper mines there. And as I'll tell you, there are also many other copper mines throughout western North America. And wherever there's water at these abandoned mine sites, Mimulus has colonized these sites and has adapted to copper. We're just beginning to explore the interactions between Mimulus and its microbes, this just shows that Mimulus has AM fungi, and so this is likely to be a very interesting avenue of research. I should say that within this complex there's an enormous amount of sequence diversity. Any 2 Mimulus accessions from this complex are typically as divergent as human and orangutans. So these are, and yet they're interfertile and you can do crosses. Population structure is often very large, a lot of local adaptation to habitats. So I'll just tell you a few quick stories here. The first involves the difference in life history and salt tolerance between Mimulus-Vitis plants that live here on the Pacific coast. These are perennials. They flower late in the season, in the late summer and fall. Very large, robust plants. You can see when they're sprayed with the salt spray, they still persist despite that heavy salt content. If you go over the edge of the Coast Range inland, you find, of course, a very different habitat. This is the classic Mediterranean habitat of California and western US, where plants germinate in the fall, winter, or spring in the very wet seasonal rains, but then have to germinate and grow and flower and reproduce before the summer drought hits. And here you find annual populations that live fast and die young, reproduce before May or June. These types of habitats are often separated by just a few kilometers. So here's the coastal site and inland, an annual site. The plants are phenotypically very different even when you bring them in the greenhouse, the annual being much smaller than the perennial and more rapidly flowering. And any of you who live here know that this environmental gradient is enforced by the coastal fog, which keeps the coastal populations wet year-round, whereas the inland ones suffer from the drought. We're having issues with PowerPoint here, but essentially what we've done is we've collected now coastal, this is California and Oregon and Washington right here. A beautiful map, as you can see. And what we have are collections of the perennials in red and the annuals in blue, taken from just north of LA all the way up to British Columbia. What we've shown is that these races, ecological races, are diverged at a genome-wide level from each other. And what we've also shown is that they are locally adapted to their populations. Megan Hall, who's actually in the audience, did this cross between, as you can see, this Cascade Oregon population and this coastal perennial in the Oregon sand dunes, and showed that these two populations could not really do very well at all in their non-native habitats. And David Lowery has since shown the same in populations down here in California. What you see is that when you transplant the coastal seedlings into the inland habitat, they grow slowly and try to get bigger and bigger, but then they get fried by the summer drought before they can flower. This is the only plant out of several hundred that flowered, and the day after it flowered, it died. It's a very sad story. Whereas the inland ones, annuals, are perfectly happy and can reproduce as they normally do. On the coast, what you see is that the perennials can get very large and produce masses of seeds, 100 times more seeds in their lifetime than the inland annuals planted on the coast. That difference in life history obviously suits the coastal perennials fine, but as I said, the perennials are also salt tolerant. You could show this in the lab just by taking plants from the two populations and spraying them with saltwater spray, and the inland annuals die rapidly. So we'd like to know, what's the genetic basis for this difference? Megan Hall was the first one to investigate this using a QTL study by crossing inland annual to the coastal perennial. What she noticed was that very early on, just looking at the F2s from this cross, you see the recovery of plants that look almost as extreme as the two parents, suggesting a simple genetic basis to this. And when she did a very laborious QTL study before we had our fancy markers, she was able to show that there were two, and this is a difficult slide to see, there were two genomic regions that contributed to most of the difference in phenotype in terms of morphology and life history between these two. These two QTLs, both on linkage group 8, explain each about 20 to 30% of the difference in flowering time and morphology. David Lowery has since investigated the genetic basis of salt tolerance, and there are two major QTLs and a third that I'm not showing, actually a fourth as well, but three somewhat major ones. And so we're beginning to investigate the molecular basis of these traits. When we first got the genome assembly, we were thrilled because this is, for example, one of those regions on linkage group 8 that affects flowering time. This is a region of about 25 centimorgans, and it corresponds to about 1.5 megabases in the genome. This implies that in this region, it's about 50 kb per centimorgan, so we could easily, using fine mapping methods, ideally, figure out which gene it is. And we see this throughout the genome. When you look at gene-rich scaffolds, the sort of average recombination per centimorgan, or number of kb per centimorgan, is on the order of 50. This is despite a genome-wide average of about 200, suggesting that these repeat-rich regions have really reduced recombination. So when we started looking at the scaffolds that overlie these QTLs for flowering time, morphology, and salt tolerance, there are a number of very interesting candidate genes in those regions. We started to map the first flowering time QTL on linkage group 8, and normally that's a region, as I said, of about 25 centimorgans. What we found when we started to use our new genetic markers is that in a cross between the coastal and inland form, there was essentially no recombination. And we've since shown that this is due to an inversion difference between the coastal and inland races, perhaps tying together more than one adaptive gene, co-adapted gene. And it turns out that this rearrangement is associated with life history throughout the whole complex, so that perennials, whether they be on the coast or even here in some of these hot springs in the Great Basin, all have this one particular QTL and genome rearrangement, whereas the annuals, not just the inland annuals right near the coast, but also even some of these self-fertilizing annual species like Mimulus-Vitis all have the alternative arrangement. So we're not really going to be able to use forward genetic approaches to find that gene, but when we start to look at the other QTLs, we're much more successful. So here's QTL number 2 on linkage group 8 that affects flowering time and morphology. This is a manageable scaffold that has nice high recombination rates. There's a beautiful candidate for flowering time and morphology that we know about from work in Arabidopsis and other plants. This is involved in gibberellic acid. The prediction is that because GAI normally represses flowering time in Arabidopsis, that GAI should be more highly expressed in the coastal plants that are late flowering and as opposed to the inland plants. Now we're just obviously in the very early stages of this, but the results are very encouraging. Here you have the inland annuals that have very low expression of this gene as compared to the coastal perennials that have much higher expression, consistent with the idea that this is causing it, but certainly not at all definitive. We're doing a lot more work on that. So I've shown you how these Mimulus plants often adapt to differences in soil moisture by shifting their life history and flowering time, but they can also become more drought tolerant through other physiological means. So for example, by minimizing water loss to avoid dehydration when they're exposed to these hot habitats, and perhaps are more water use efficient. We've begun to explore this in a number of different populations, and I just want to show you one quick result here by comparing the annual form of Mimulus-Vitis to its self-fertilizing species, Mimulus-Vitis, that often flowers even earlier than the annual and is even more drought tolerant. It's certainly a lot more drought tolerant. You find it in drier sites than Mimulus-Vitis, even when they grow in the same habitat. And what we've shown is that Mimulus-Vitis is more water use efficient, and this is true whether you measure it using carbon isotope or other measures of transpiration. And we've identified 2 regions that have large effects on water use efficiency, and these are on linkage group 7 and linkage group 13. Each explains about 30% of the difference between those 2. What's interesting is that at least for one of these regions, we see that it seems to be correlated with the water use efficiency at these QTLs seems to be associated with differences in stomatal density. And you can see here that Mimulus-Vitis genotypes at this QTL have a much higher density of stomates than Mimulus-Vitis do. And interestingly, in this region for this water use efficiency gene, again, we can find some candidates. Obviously, there are probably lots of candidates in here, but this one in particular is interesting because in Arabidopsis, that gene seems to work in a pathway that does influence both stomatal density as well as water loss due to dehydration. Okay, so those are just a couple of examples of some of the traits that we're beginning to look at. There are lots of other things that we are pursuing and other labs are pursuing. For example, there are 2 species that are restricted to very high altitudes and different habitats. Here's Mimulus-Vitis that lives on granite outcrops in the Sierras, and this is Mimulus-Tilingii, which is only found really above 10 to 12,000 feet. But what I'd like to tell you about at the very end of the talk is some of the work that we've been doing with some of these soils. I mentioned the copper-tolerant populations of Mimulus-Vitis. These are mines in Copperopolis. It's been known since the 80s that Mimulus-Vitis is able to live on these soils to a large degree because of a single locus that confers copper tolerance. The interesting thing about these mines is not only is Mimulus-Vitis there, but you also find this self-fertilizing derivative of guttatus that is only found on 2 mines in Copperopolis and nowhere else in the world. And it is tolerant due to the same gene, but it is also much more tolerant to copper than Mimulus-Vitis and has a number of other adaptations that allow it to persist in these very dry, harsh, nutrient-poor sites. So we decided to collaborate with Mark McNair and try to positionally clone that major tolerance gene. And what we were able to do is very quickly map it to a position, a very fairly finely defined position on linkage group in Mimulus-Vitis. We would basically get DNA from plants in a mapping population that Mark McNair had and genotype them. When we got the sequence assembly, we were very, very hopeful, but that lasted only a few minutes because we realized that this is one of these regions that is not particularly well assembled, in part due to these repeats. So this is a great example of how hopefully we should be able to, with this genetic mapping of the scaffolds, begin to figure out what scaffolds reside between the scaffolds delimited by our genetic markers that flank the tolerance locus. Now, the other nice thing is that throughout Western North America, there are a number of these copper mines, and on each of them, if there's any water around there, Mimulus-Vitis has colonized those sites and again has become tolerant to copper. And we're now in a position to see if that same locus is responsible for tolerance at these other sites or if other novel mechanisms are involved. I mentioned serpentine soils. We're just beginning to work on serpentine, as are some other labs. Serpentine, in case you don't know, are low in available calcium, very high magnesium content, high concentrations of heavy metals like nickel and cadmium. They also have very low water holding capacity and very poor nutrients. So adapting to these environments is a tricky feat, but Mimulus has done it over and over again. The interesting thing is, like the case with copper, we see not only Mimulus-Vitis residing on serpentine from California up through the Pacific Northwest, but in some regions you also see little endemics that are derived from Mimulus-Vitis. This is Mimulus nudatus from Lake and Napa counties, just north of here, that is only found on serpentine soils. You could go to the Sierra foothills and to the serpentine soils there and find Mimulus pardalis, which is a self-fertilizing species only found on those serpentine soils. We would like to know what's the basis of the serpentine tolerance, as well as some of the unique features of these endemics. Okay, but what I've shown you are a number of different ecologically important traits and how we can use forward genetic approaches to begin to attack them. But I've shown you how we've been doing this sort of one cross at a time, one QTL at a time. And obviously this works, but it's not particularly efficient. It's not particularly scalable. It's a lot of work for each one of these QTLs. And what we would like to do is develop a much more powerful system that would enable us, as well as anyone in the research community, to really go after genes of ecological importance that have to do with how these plants adapt to changing environments, abiotic as well as biotic conditions, traits that we haven't even imagined yet. And so what we're wanting to do is we currently have close to 1,000 inbred lines that we've extracted from well over 100 populations and closely related species, all of which are interfertile. These represent a tremendous range of adaptive variation within this group. What we would like to do, if you can see the partial words over here, is select some of the most extreme and use them in what is known as a collaborative cross, where we basically intermate them all so that the resulting hybrids and resulting recombinant inbred lines are mosaics of all of these diverse founding parents. And then these are permanent mapping resources that can be used by anyone, and they're also extremely powerful in being able to very rapidly map QTLs down to, if not a single gene, to very small genomic regions. And so this is what we're working with at the moment to try to develop this as a much more powerful platform for our future ecological genomic studies. Okay, well, thank you very much for your attention, and I'd like to acknowledge that the work in our lab has really the result of lots of effort. These are the current students and postdocs in the lab. I mentioned David Lowry and Megan Hall, who is recently graduated from the lab, initiated the work on the coastal plants. Kevin Wright is working on the copper tolerance plants. Carrie Wu and this very talented undergraduate Laura Nutter have been working on the water use efficiency project, as well as support from many other collaborators, especially JGI and many other people, support from NSF and NIH. Thank you for your attention.
Thanks, John. Any questions?
So, I don't know about plants that much, but is Mimulus just exceptional and that's why you study it, or are there many other examples of plants that can be this flexible and that's just the power of plant genomes?
My personal feeling, well, there are a number, so shall I repeat the question? Oh, okay. The question was, is Mimulus unique, or are other plant systems this diverse? The answer is probably the latter. One thing that Mimulus has going for it is that it lives in western North America, which is one of the most ecologically diverse areas, at least in this part of the world. There are lots of other genera that I can think of that have adapted to very diverse habitats. What I think Mimulus has going for it is that it's been worked on really quite continuously since the 1940s and 1950s by ecologists, starting with work at the Carnegie Institute all the way up to now. And so there's a fairly large community that has developed around it, and so they've really shown that this diversity exists and that you can cross these things and do the genetic studies. But my guess is there are lots of other species that have similar levels of diversity. What's nice is that they're not all reproductively isolated from one another, which isn't always the case.
2 months generation time.
And 2 months generation time, big selling point. That's right, even the woody ones. Eddie.
I sort of have a mirror image question. I've not heard of plant geneticists that did not try to beat me over the head with the phenotypic diversity of the plant they worked on.
Yeah.
And so can you ever use that as people identify QTLs and genes contributing to specific phenotypes? Can you use it between plant species?
Absolutely, and I think a lot of the examples that I showed here today show how the work that we're doing in Mimulus and asterid has benefited tremendously from the work in Arabidopsis and other species. And I think that the more we investigate ecological genomics adaptation in diverse plant groups, the more there's going to be synergy. It's got to be true. Obviously, plants share a great number of genes in common across the whole phylogeny, and so I think this is where the power of comparative approaches will really play out. Well, I don't know. Dan's in charge.
I was just wondering, within the asterids, what plants that can be used for food, feed, fiber, and fuel is Mimulus most closely related to?
Well, I showed you the example of microsynteny with Solanaceae, tomatoes, potatoes, plants in that group. Mimulus is eaten. Down in South America, it's a little green that has been used by the native people there for centuries. But in terms of major crops, I guess I would say Solanaceae. And then, of course, in asterids, you have things like sunflower, lettuce, things like that in the other group of asterids. Coffee is not too far away. That's a big one for me.
I was curious what the natural outcrossing rate was in Mimulus, the species you're looking at, and whether you see inbreeding depression when you create these
Yeah.
nominated.
That's an excellent question. It's certainly one I glossed over. Mimulus-Vitis itself, as I said, is largely outcrossing, although populations do vary. And what I mean by that is that typically 80% of the seeds produced by any one plant in a typical population will be outcrossed. Maybe 10, 20, 30% self-seed. Nonetheless, the guttatus itself has a lot of genetic variation. Whenever you have a lot of genetic variation within populations and you're largely outcrossing, there's a lot of inbreeding depression. So you do lose lines. And that's actually something that people have studied a lot. It's what I studied a lot in the 90s, for example. There's a fair amount of genetic load, but nonetheless, because these things are so prolific and you can easily grow so many in the greenhouse that you can tolerate a little bit of line loss, or sometimes even a lot of line loss, and still end up with quite representative inbred lines that are quite healthy. But as I mentioned, there are also a lot of these little endemics are self-fertilizing specialists for certain habitats, and they really don't suffer inbreeding depression. Yeah.
Hi, John. I just wanted to say, great to see another plant coming online.
Great.
And my first question is, I got two. First one is, when is it going to be made available for the public to start using, if you know?
Well, I think we're waiting until we finish the annotation. Maybe Dan can address this, but I think we're shooting for something like, I don't know, sometime this summer.
My second question is...
I mean, so I should point out that the reads themselves are available, through Trace Archives, but that doesn't, help you a great deal. But the rest of it should be out quite soon.
Excellent. My second question is, when it comes to the synteny that you were showing between Mimulus and Gray, one of the other interpretations is that they share this more ancient genome duplication event that's only been seen in the roses called paleohexaploidy.
Right.
Can you talk about that in terms of how confident you are that this is an asterid-specific genome duplication event versus one that's shared between the roses and the asterid?
So I think Dan's probably the one to answer that, but as I understand it, it's quite well supported. But Dan, do you want to add to that?
Yeah, the data is just really preliminary, but it looks like that. So, just sort of crudely speaking, grapes have 21 chromosomes. That's 3 times an ancestral 7 of the pre-hexaploidy. And then it looks like each of those 7 is duplicated twice in Mimulus.
Any other questions?
Yeah, well, at least I have one. Thanks, wonderful. I'm curious about, through your insights of all these adaptations, about correlation in various time frames necessary to result in this speciation. It strikes me with all the, possible connections with habitat types there, and you must think about this issue of time frames. When did this occur, and how much time is necessary? And given a lot of what you know about the lack of let's say cross-fertilization among these, does that give you some insight into the time frames?
Well, I think it's probably fairly old. I mean, what I mentioned was there's really very large sequence divergence between any two populations, or even between individuals within the same population. So for example, if you align coding regions between the inland plants and the dune plants, or between the inland guttatus and the self-fertilizing Mimulus-Vitis, and you just look at the percent sequence divergence between them, it's on the order of 2.5% on average. So that suggests that this molecular diversity, at least, is very old and has been segregating for a long time. I think that they're basic, but other than that, we don't have any good estimates of dates or oh, there you are. Some sense of the time frame. I should say that we really have not done much yet in the way of population genomic analyses, and part of that is that we've been focusing on these specific crosses, but we're now moving into a phase where there's a big effort that's just starting to really do a comprehensive phylogeographic population genetic study of the entire complex to see what extent there is sort of genome-wide isolation between these ecological races, for example, and perhaps that will give us a better sense of the time frame. Yeah.
Thanks a lot, John.
Great.